Eating disorders are a class of mental illnesses characterized by abnormal eating behaviors like too much or too little food, obsessions with food, body weight and shape, usually resulting in drastic changes in the person's size, shape, and overall physical and psychological health. Now, there's a whole bunch of different eating disorders out there. However, when most people think about eating disorders, they think of the big two, anorexia and bulimia, and to a lesser degree, maybe binge eating disorder. They are, after all, the most prevalent, and so, as the consequence, are the most widely studied. So in this video, I'm going to go over some of the neuroscientific evidence and research that has been done analyzing the brains of people with eating disorders. We'll start with anorexia nervosa. Taken from the DSM-5, diagnostic criteria for anorexia include reduced calorie intake and loss of weight, intense fears of gaining weight, and distortions in self-image. There's a whole bunch of identifiable symptoms of anorexia, some psychological like low self-esteem and antisocial behavior, and some very clearly related to brain and body changes, which we will talk about a bit later, like depression, fatigue, poor health, and if taken far enough, the cessation of hormone production. If a person has anorexia for long enough, then they run the risk of osteoporosis, cardiovascular disease, diabetes, thyroid disorders, gastrointestinal disorders, infertility, and the development of other psychopathologies. Anorexia also happens to be the mental illness with the highest mortality rate, so pretty serious business. Bulimia nervosa is an eating disorder characterized by binging and purging, or the excessive rapid consumption of food followed by getting rid of the food, usually by vomiting. Binge eating and the compensatory behaviors both occur, on average, at least once a week for 3 months. Many of what we've talked about in reference to anorexia apply to bulimia as well, with the added bonus of rotting teeth, excessive heartburn, and bursted blood vessels in the eyes from vomiting. Now, how exactly prevalent these disorders are is a bit tricky, depending on who you ask and where you ask and how you ask, but I'm going to stick with the numbers given by Hans Hoek, a researcher out of the Netherlands, and in any given year, anorexia hovers around 0.3% of the population of women, and bulimia around 1%. Here's a chart I made with data taken from the World Health Organization in 2013 and a cross-national survey of binge eating disorder, which I'm not going to talk too much about, and bulimia. And here you can see there's lots of variation between countries, and I've highlighted some of the top spots. Over the course of this video, I'm going to be covering 3 main differences in the brains of people with eating disorders like anorexia and bulimia. One molecular, one emotional, and one cognitive. As well, I'll also cover some of the neural correlates of more specific behaviors like binging and purging. First, I'm going to talk about each individually, and then I'll do that in order, and then at the end I'll attempt to bring them all together into a kind of cohesive story of the disorder. After that, I'll finish with a theory of the development of eating disorders and try to explain why people might actually get them in the first place. So, here we go. One of the major differences seen in brains of people with eating disorders is in the neurotransmitter serotonin. And here we see a very general outline for 2 major neurotransmitters, serotonin and dopamine, both of which undergo significant changes in the brains of people with eating disorders. But for now, I'm just going to cover serotonin. Serotonin is synthesized in a cluster of tiny regions called the RAF nuclei, located in the brainstem, which we see here in this model of the brain at the bottom. This small set of nuclei sends signals containing serotonin pretty much, well, everywhere. And so its function is very extensive because it spreads all over the brain, and so it's related to things like mood, cognition, memory, sleep, but most importantly for our conversation, eating. The molecular precursor to serotonin, or the thing that serotonin is made out of, is tryptophan. And the only way to get tryptophan is through your diet. So if your diet is bad, well then, you won't get enough serotonin. Now, it's not that having a bad diet leads to less serotonin, which then leads to an eating disorder. In fact, it's somewhat the opposite, that eating disorders are thought to be disorders of too much serotonin, and that restricting eating might actually be a way of controlling that. dysfunction or malfunction in the serotonergic system leads to a number of behavioral pathologies, and there's extensive evidence indicating that the level of serotonin receptors, or the molecules that serotonin binds to produce effect on your nervous system, change drastically with eating disorders. There are many different types, but I'm going to focus on the two primary ones, the 5-HT, which is just short for serotonin, the 5-HT1A receptors, which are most often inhibitory, meaning that when serotonin binds to it, there will be a general decrease in neural activity. The 5-HT1A receptors increase in prevalence by about 50 to 70% in certain areas of the cortex compared to healthy people, specifically in the orbitofrontal cortex, parts of the parietal cortex, and the cingulate regions. Mesial temporal regions, so things like the hippocampus and the amygdala, as well as the dorsal raphe nuclei itself, where serotonin is synthesized. 5-HT2A receptors, which tend to be excitatory, resulting in more neural activity, tend to be decreased in many of these same regions. Now, what does all this mean? Well, disrupted neurotransmitter function ultimately leads to disrupted neural activity in the brain regions that it's disrupted in. The relationship between various different serotonin receptors and behavior is pretty complicated. Studies comparing serotonin function during and after the diagnosis and treatment of eating disorders indicates that these serotonin receptor differences last well after the disorder has been dealt with, indicating a strong influence and role that genetics have to play in the disorder. Now, I mentioned several brain regions that were affected by this. Now, along with these serotonergic specific changes, there have also been several functional imaging studies that have been done using fMRI and PET to determine what brain regions are affected in those with eating disorders and how they contribute to behaviorally relevant and functional circuits. Now, the most obvious issue in people with eating disorders is their relationship to food. And there exists a series of brain regions that we have that will judge, place value on, and elicit emotional responses to food. Most of these regions I'm going to be talking about in this emotional value system have been implicated in serotonergic-related abnormalities like the ones I just talked about. There are three regions in the cortex which are almost always found to have abnormal activity associated with eating disorders. These are the anterior cingulate, the insular cortex, and the orbitofrontal cortex. We see the anterior cingulate cortex here. Now, there are different components, different component pieces to the cingulate, and because both receive and send information to and from different places, are thought to have different functions. The anterior cingulate cortex is a very popular brain region of study and has a lot of functions associated with it, such as error processing, performance monitoring, action selection, and value judgments. Now, these are, of course, neuroscientific terms, so we'll have to break this down a bit into something simpler. In the broadest sense, we might be able to say that the anterior cingulate cortex does two general things. It takes in information about possible options, determines whether or not they are good or bad, and then picks one. This is something referred to as a comparator or conflict monitor. Secondly, it is an important part of a larger system that determines the emotional value of the things we interact with. When it comes to eating, it is thought that the anterior cingulate cortex will receive information about food, determine how valuable the food is, and then make decisions about whether to engage with it or not. Or that is to say, is the food good or bad, and should I eat it or not? And it does this through extensive connections with the insula and the orbitofrontal cortex, which we'll talk about right now. Now, it's no surprise that the insular cortex, usually just shortened to the insula, is involved in eating, as one of its primary functions is in taste, and so it sometimes gets the name insular taste cortex. However, there are a number of different functions, way more advanced functions, that have been associated with the insula. The insula has been implicated in a wide variety of functions important to emotion, salience, sensory integration, and the sense of self, which we'll talk a little bit more about later when we go over body image and anorexia. To say the least, it's a very complicated structure, and there's many theories and models of its function. But for now, the insula's role in this emotional value system we're talking about seems to be to attach emotional significance to incoming sensory information and bodily states and produce emotional feeling itself, or at least play some critical role in the production of emotion itself. So some possible examples of the insula's emotional function might be in seeing a disgusting, rotten piece of food and then producing a feeling of disgust in you, and then you avoid it. Well, the insula might play a role in integrating the information about the food, producing the feeling of disgust, as well as sending signals to other brain areas involved in moving you away from the disgusting food. So with connections with other limbic emotional structures like the Anterior Cingulate cortex and the amygdala, it's critical for emotional production. With its connections to the prefrontal cortex, the insula is critical for awareness of emotion and thought, and connections to the striatum influence motivations and positive reward. It's a very complicated part of the brain, so for now, we'll simplify things and call it an integrator of emotionally relevant information and producer of emotional states. Similar to the ventral portion of the anterior cingulate, the orbital frontal cortex has been shown to have a role in evaluating and judging things. And given its extensive connections with the insula and the anterior cingulate cortex, it's not surprising that it has a role in making emotional value judgments that guide decision making. And we can see here where the orbital frontal cortex is. There is a specific pattern of brain activity seen in these three cortical areas when someone interacts with food. And this pattern appears to be greatly reduced in people who have eating disorders, highlighting some kind of dysfunction in this emotional value judgment system. Alongside with these cortical regions, subcortical, or below the cortex, brain regions are also important in the production of emotional states and movements, namely the amygdala and the striatum. The amygdala are a pair of almond-shaped structures that are thought to code for emotional memories. When you engage with an object or an event and something happens that produces an emotional response like pleasure or pain or fear or anger, the amygdala will store memories of the emotional nature of the thing or the event. It's perfectly set up to do this as many of its inputs are from sensory areas, and the major point of its output is the hypothalamus, or the center of hormonal production and bodily regulation. It's the amygdala's connection with the cortical regions I've talked about so far that make it a necessary component in the production of emotional responses, as well as learning about the emotional significance of food. With eating disorders, the amygdala is more active when someone looks at or interacts with food, and as a result produces more anxiety and stress. Finally, there is the striatum. The striatum is a cluster of small nuclei with a number of functions, although, just like the cingulate cortex, can be divided up into a dorsal and ventral component. This distinction is made mostly by the type of inputs and outputs that each component receives and sends, and so ultimately both have distinct, unique functions. The ventral striatum, which is also called the nucleus accumbens, plays a critical role in reward, reinforcement, and motivation. It's one of the primary reward or pleasure centers of the brain, if that's what you want to call it, and through its use of dopamine processes information about what is pleasurable and what is not. We can easily see how food would fit under this category, and there's plenty of evidence that there are dopamine receptor changes in the striatum of people with eating disorders. Collectively, all of these brain structures can be conceptualized as a ventral limbic system. The ventral limbic system has two main proposed functions. It identifies emotional significance in things in its environment and produces affective or emotional states in response to them. It also regulates and produces behaviors in response to that stimulus. So, not only does the system attach value to a thing, but it drives your behaviors towards the thing, sending signals to brain areas responsible for approaching the object and movement as well. It also produces positive and negative emotions. So in eating disorders, alterations in serotonin receptors have been found in many of these same areas I've just talked about. fMRI studies have shown decreased activation in cortical regions that I just talked about, increased activation in the amygdala coinciding with increased anxiety, and abnormal dopamine related to activity in the striatum. With this neurobiological model, it appears that people with eating disorders are inappropriately judging the value of food and producing intense, negative emotional states like disgust and anxiety in response to it. Not only this, but changes in dopamine responsivity in the striatum appear to change how rewarding these people find food. And this leads us to our next topic. One of the primary symptoms of bulimia is the binge, eating excessive amounts of food in a very short period of time. The behavior of abnormal binge eating is going to take place largely in the reward system and involve dopamine. Now, most people have a fairly simple but accurate enough understanding of dopamine. Something pleasurable happens and dopamine is released in your brain. This has a reinforcing effect on your behavior, changes the structure of the neural connections, and can result in things like addiction. So it's probably no surprise that binge eating results in large releases of dopamine in the nucleus accumbens. Binge eating has been shown to increase expression of the D3 receptor, a type of dopamine receptor, and decreases the expression of D2 receptor, another form of it. Molecular changes identical to what happens in drug addiction. It has also been shown that people with bulimia have overall lower levels of dopamine. And on top of this, starving, in anybody for that matter, has been shown to increase the dopamine response to food when you get it. And so we were left with a situation in which the reward value of food becomes greatly heightened. But this doesn't explain why people don't just, eat all the time and get fat. The other half of bulimia is the purge, or getting rid of the food after. Well, when it comes to binging and purging, it's possible that because of the reinforcing nature of dopamine, people will become addicted to binging. However, food is a necessary precursor to serotonin, like we talked about, meaning that the more you eat, the more serotonin you produce. Serotonin receptors are expressed in several of the emotional areas, like the amygdala, and can produce anxiety. So in some people, a vicious cycle might begin. Eat food, get a dopamine rush that feels good. Food leads to serotonin. Serotonin rush produces the feeling of anxiety. So how do you solve this problem? Well, you eat all the food in a binge, and then you get rid of all the food in the purge. However, there are many other factors at play when it comes to binge eating. The hypothalamus sends signals to the nucleus accumbens to influence serotonin activity, and there are many other neurotransmitters like acetylcholine and opioids that are also thought to play a role. Binge eating is also closely related to a more cognitive process called self-control, which leads us to our next discussion about the prefrontal cortex. Now, we've already talked a little bit about the prefrontal cortex in terms of the orbital frontal cortex, but another important area of interest with regards to eating disorders is something called the dorsolateral prefrontal cortex, or the DLPFC for short. Some general functions of the DLPFC are thought to be planning complex cognitive behaviors, social behaviors, and decision making. And it does all of these things through extensive connections with several other brain regions that all give it the necessary information to do these things with. It has extensive connections with the orbital frontal cortex and the striatum, for example. Now, the physiology and computation behind how it actually does this was beyond the scope of this video, but with planning and decision making comes self-control and the ability to do one thing and not another, as well as purposefully restrict one's own behavior when given whatever social context they happen to find themselves in. With regards to self-control, it is thought that serotonin activity in the prefrontal cortex modulates inhibition and control. Too much activation of either one of the two main receptor subtypes can lead to a disorganization and the loss of self-control, or maybe even resulting in a binge. Or in the opposite direction, too much control and a strict regulation of eating behaviors can lead to the type of over-control that we see in the diets of those with people with anorexia. The striatum is, of course, highly connected with the prefrontal cortex. And many of the brain regions that both send signals to and receive signals from the prefrontal cortex are also serotonergic and affected by this disorder. It's widely known that those with eating disorders tend to view themselves as bigger than they actually are. Now, is this just, paranoid ramblings and baseless overstatements, or do these people genuinely believe that they are, in fact, bigger? Do they see themselves as literally bigger? Is there some disconnect in their perception of themselves and the perception of other people? Well, there's some evidence to indicate the latter. Imaging studies have identified a pathway in the brain dedicated to processing body shape information, and it includes the extrastriate body area, the fusiform body area, the inferior parietal lobe, the dorsolateral prefrontal cortex, like we just talked about, and distinct parts of the parietal lobe. Sometimes called the body schema network, these areas seem to be necessary for perceiving body shape of others and of yourself. So first we'll start with the extrastriate body area and the fusiform gyrus. Now, I have other videos which go over the fusiform area in much more detail, but to simplify things for here, the extrastriate and fusiform region encodes information about body and face, respectively. Information from the visual cortex encodes all the visual information that your brain receives, but when whatever it is we are looking at resembles the shape of a body or a face, the signal gets sent to these regions to specifically make sense of and encode faces and body shapes. A second system involves the prefrontal cortex again and parts of the parietal lobe. The inferior parietal cortex is a region that, when damaged, can produce specific types of neglect symptoms or a denial of illness, and is also related to the movement of the body in relation to the space around you. It is thought that signals from the visual cortex go to the prefrontal cortex and the parietal lobes to direct attention and the perception of the body. And so, one really interesting thing that you can do is that you can take people with anorexia, and you can take, relatively healthy people, and you can present to them images of themselves as well as distorted images of themselves. So, it's like a picture of you and then someone photoshops a bunch of it. And what will happen when you look at images of you is that a specific type of activity in your brain will happen, involving the DLPFC and the extrastriate cortex, and also the insula. Now, when you take that image and you distort it with Photoshop, there'll be, a very different type of activity. This doesn't happen in people with anorexia, indicating that how people with anorexia process images of their own body is very different from how healthy people do, and that they cannot make the distinction between a distorted body shape and a regular body shape. So, throughout this video, I've talked about several neurobiological differences that occur in people with eating disorders. The first is changes in serotonin receptors. The changes in serotonin receptors happen in very specific brain regions that have been theorized to be part of behaviorally relevant systems. The first is the ventral limbic system that attaches emotional meaning to things, produces emotions themselves, and orients your behavior to or away from the thing that you're interacting with, depending on whether it's a positive or negative emotion. The other system is a visually guided system about body image or body schema. Altered activity in this system has thought to rely on an inaccurate self-perception, leading some to think that they're fatter than they actually are. Within this framework, I covered how unstable dopamine responses can lead to food addiction and binge eating, as well as prefrontal cortex impairments that can lead to too much or too little self-control. Now, with all this in mind, we're now going to talk about theories as to why people and how people get these disorders in the first place. Some research indicates that many of the personality traits and other psychological illnesses that occur prior to an eating disorder diagnosis in most of the young women who end up with the diagnosis, things like perfectionism, obsessive compulsion, high anxiety, and social phobias, are linked to an already present serotonin dysfunction. Not only this, but brain imaging studies designed in such a way as to map serotonin function finds persisting serotonin dysfunction after the eating disorder has been successfully dealt with. What this means is that serotonin dysfunction is likely to be already present in a person before they develop the eating disorder and lasts well after they've been cured. This can be a combination of both genetic predisposition as well as early life stress like, childhood abuse or something. The onset of puberty and the rush of hormones that occur with it have been hypothesized to negatively impact stress hormone responses in these individuals, making them more susceptible to the negative influence of stress later on in life. A few years later, an intense level of pressure from the outside world to be thin or to look a certain way, combined with these earlier factors to produce a young woman with high anxiety and serotonin dysfunction. Some have gone to speculate that by reducing your diet and limiting and restricting your diet, you can lower the serotonin levels in your brain. That this is a way of reducing anxiety and phobia caused by the serotonin dysfunction. Too much serotonin will cause behavioral problems, so if you eat less, then you'll make less serotonin and you'll solve your behavioral problems. But the other side of that coin is that you also end up losing too much weight in the process. In this sense, eating disorders can be thought of as a kind of coping mechanism to alleviate serotonin-related stress and anxiety. Now, importantly, all of the information that I've covered here is not mine. I do not have the time or the money to run hundreds of experiments. All that I've done today is show you the results of hard work from neuroscientists over the years. This is simply my presentation. So if you're interested in learning more, then please check out the links and references that I've got in the low bar. And as always, thank you for watching, and have a wonderful day.